Welcome to the Deep Dive. Today, we're getting into one of the most fundamental challenges in all of biology.
Right. How does the cell take the information that's just, stored in DNA
The static blueprint.
and turn it into all the dynamic parts that make a cell actually live and breathe? We're talking about gene expression.
And this is going to be an extensive deep dive, starting right from the beginning, the history of what a gene even is, and then walking step by step through the actual mechanics of transcription and translation.
That's right. And our source material here gives us a really precise roadmap. Our mission is to chart that entire flow of information, making sure we verbally describe every key experiment, every molecular machine.
So you can actually picture things like, say, the title-binding protein physically bending the DNA.
Or the splicesome twisting an intron into a lariat shape, or a ribosome literally ratcheting its way down a messenger RNA. We want you to see it all.
Okay, so let's start that journey with the idea of the gene itself. It wasn't one single discovery, right? The concept was built up over time.
It really was. You had Mendel, who first proposed these discrete elements for traits.
Then Boveri and Sutton came along and correctly figured out that these elements were physically located on chromosomes.
And of course, Watson and Crick gave us the chemical reality. A gene is a segment of DNA. But the big question was, what does a gene do?
And the first major clue came way earlier than you might think.
It did. In 1908, a British physician named Archibald Garrod was studying what he called inborn errors of metabolism.
And the classic example is alkaptonuria.
It's the perfect example. Garrod had patients whose urine would turn black when it was exposed to air.
Which must have been pretty alarming.
I would think so. He figured out this was happening because a specific chemical homogentistic acid was building up in their bodies.
And a healthy person would normally break that down.
Exactly. So Garrod made this brilliant deduction. These people must be missing a specific enzyme that's supposed to oxidize that acid. He connected a genetic defect directly to a missing enzyme and a specific medical condition.
That is a huge conceptual leap. A gene makes an enzyme. But that idea didn't really catch on for a few decades.
Not until the 1940s with Beadle and Tatum and their work on Neurospora, the pink bread mold.
They designed this incredibly elegant experiment to prove the connection.
They did. So you have to understand, wild Neurospora can grow on what's called a minimal medium, just some sugar, salts, and a vitamin.
Because it can make everything else it needs. All the amino acids, other vitamins, all by itself.
Right. So Beadle and Tatum's plan was to break that ability. They zapped the fungal spores with radiation to cause random mutations.
And then they looked for mutants that couldn't grow on that minimal medium anymore.
Exactly. So if a mutant spore couldn't grow, they knew they'd broken something in its metabolic machinery. Then they started adding things back in, one by one.
So they might add, say, the amino acid arginine to the minimal medium.
And if the fungus suddenly started growing, they knew the mutation had knocked out the gene for the enzyme that makes arginine.
It's so logical.
Yeah.
And this led directly to their famous one gene, one enzyme hypothesis.
A very powerful idea. It had to be tweaked later, of course.
Because many enzymes are made of several different protein chains.
Right, different polypeptides. So the idea shifted to one gene, one polypeptide. And even that's a bit too simple for today with things like alternative splicing.
Which lets one gene code for multiple different proteins.
So we land on our modern definition. A gene is a piece of DNA that holds the information to make either a single polypeptide chain or a functional RNA molecule.
And that brings us to the core process, the flow of information.
The central dogma. It goes from DNA to RNA in a process called transcription.
And then from RNA to protein, which is translation.
Transcription is really just rewriting the information. The language of DNA's deoxyribonucleotides is transcribed into the language of RNA's ribonucleotides,
Creating what's called the primary transcript. And in eukaryotes, the key player here, the go-between, is messenger RNA, or mRNA.
The mRNA is just a temporary copy of one strand of the gene. And using an mRNA intermediate is, well, it's a brilliant solution to a couple of problems.
First, the architectural problem. The DNA, your master blueprint, stays safe in the nucleus.
While the mRNA, the disposable photocopy, travels out to the cytoplasm where the work gets done.
And second, it allows for massive amplification.
A huge amount. You can make hundreds of mRNA copies from just one gene, and then each of those mRNAs can be used to make thousands of protein molecules. It's incredibly efficient.
Okay, so then translation happens. This is where the ribosome reads the mRNA sequence and builds the protein. But there are other critical RNA molecules involved here.
Absolutely essential ones. You have ribosomal RNAs, the rRNAs, which are the actual backbone of the ribosome. They provide the structure, and crucially, they are the catalyst.
They're the ones that actually form the peptide bonds.
They are. And then you have the transfer RNAs, the tRNAs. They are the ultimate adapters. They're the molecules that physically read the language of nucleotides on the mRNA and translate it into the language of amino acids.
It's also worth pointing out how different RNA is from DNA structurally. DNA is this long, uniform double helix.
But RNA has to do things. It has to be a catalyst, an adapter, so it needs complex 3D shapes.
And it gets those shapes by folding back on itself.
Right, creating these double-stranded regions called stems and single-stranded parts called loops. The final structure is also stabilized by things you don't really see in DNA, like GU base pairs and lots of chemically modified bases.
And those modifications aren't just for stability, are they?
No, they're critical recognition sites. They're like little flags that the cell's machinery can recognize, allowing RNAs to interact with proteins and other RNAs with incredible specificity.
And this is all before we even get to the huge world of non-coding RNAs, the snRNAs, sNORNAs, miRNAs, all these RNAs that don't make protein but instead regulate everything.
It's a total paradigm shift from the old view. RNA isn't just a messenger, it's a structural component, an adapter, a catalyst, and a master regulator. It's everywhere.
All right, let's get into the mechanics. Let's start with transcription. This is all done by enzymes called DNA-dependent RNA polymerases.
Or just RNA polymerases. And these machines can't just start anywhere on the DNA.
They need a starting line.
Exactly. They have to find and bind to a specific DNA sequence called a promoter. And they can't even do that on their own. They need help from accessory proteins.
The transcription factors.
Right. These factors are what guide the polymerase to the correct promoter to get the process started.
Okay, so let's zoom in on what happens once it gets going. The elongation mechanism. The polymerase crawls along the DNA template strand.
In the 3' to 5' direction?
And it builds the new RNA strand in the 5' to 3' direction.
And to do that, it has to locally unwind the DNA, creating what's called a transcription bubble.
Oh, how big is that bubble?
It's pretty small, only about 15 base pairs long. And inside that bubble, the new RNA strand forms a very short, temporary hybrid helix with the DNA template, maybe 9 base pairs long.
And that little hybrid helps hold everything in place.
It does. Then, as the polymerase moves on, the DNA zips back up behind it.
And the whole process is basically irreversible. How does the cell make sure of that?
Through some clever energy coupling. The polymerase uses ribonucleoside triphosphates, or NTPs, as building blocks. When it adds one to the chain, it cleaves off 2 phosphates linked together, which is called pyrophosphate, or PPI.
And then another enzyme immediately comes in and destroys that PPI.
Exactly. An enzyme called pyrophosphatase chops the PPI into 2 individual phosphates. That second reaction releases a ton of energy, and it's that energy release that effectively pulls the whole polymerization reaction forward and makes it impossible to reverse.
And accuracy is amazing. How does it make sure to put an A opposite a T and a G opposite a C?
It's all about geometry. The active site of the polymerase is shaped so that only a nucleotide that forms a perfect Watson-Crick base pair with the template will fit. Anything else is the wrong shape and gets rejected.
These polymerases are also incredibly processive, meaning they stay attached for thousands and thousands of bases. But they do sometimes hit roadblocks.
They do. They can pause or even backtrack if they encounter damaged DNA or tricky sequences. And there are other proteins, elongation factors, that can come and help them get through it.
And they can also proofread, right?
They have a basic proofreading ability. If the enzyme accidentally adds the wrong nucleotide, it can back up, snip out the mistake, and try again. It's not as good as the proofreading in DNA replication, but it still significantly boosts fidelity.
What's incredible is that scientists can actually watch a single one of these enzymes at work.
It's mind-blowing nanotechnology. You can, for example, stick a single RNA polymerase molecule onto a glass slide.
And then you give it a piece of DNA that has a tiny fluorescent bead attached to the end.
Right, and then you just watch the bead with a powerful microscope. As the polymerase transcribes the DNA, it pulls the DNA through, and you can see the bead moving. You can measure the speed, see if it pauses.
And you can even measure the force it generates.
You can. Using something called an optical trap, which is basically a focused laser beam, you can grab onto that bead and pull back. You can measure exactly how much force it takes to stall the polymerase.
And the answer is?
They are incredibly powerful molecular motors. They generate forces several times greater than a myosin molecule in your muscles. They are physically plowing their way down the DNA.
Okay, let's contrast this process in bacteria versus eukaryotes. Bacteria are much simpler.
Much simpler. They have just one type of RNA polymerase. The main part is called the core enzyme, made of 5 subunits.
But the core enzyme on its own is basically useless for starting transcription.
It's blind. It will bind to DNA randomly, but it can't find the promoter. To do that, it needs to team up with another protein called the sigma factor.
And together, the core enzyme and the sigma factor make up the holoenzyme.
Exactly. The sigma factor is the guide. It tells the core enzyme where to start. Once the holoenzyme binds the promoter and starts making the first 10 or so nucleotides of RNA, the sigma factor usually just falls off.
And the core enzyme then takes off down the gene in its high processivity mode.
Right, and the bacterial promoter itself has a very clear structure. There are 2 key sequences that sigma recognizes.
The -35 element and the Pribnow box at nexus 10.
Exactly. The Pribnow box is especially important for pinpointing the exact start site. The sigma factor binds there and actually flips 2 of the DNA bases out of the helix, which starts the melting process.
And bacteria have this brilliant system of using different sigma factors to control whole sets of genes at once.
It's incredibly efficient. So the main housekeeping sigma factor is called sigma $70, and it recognizes most of the cell's promoters.
But if the cell gets stressed, say by high heat,
It quickly makes a different sigma factor. This new one ignores the normal promoters and instead directs the RNA polymerase to a whole new set of genes, the heat shock genes, that the cell needs to survive.
and how do they stop? How does termination work in bacteria?
2 main ways. One involves a protein called Rho, a ROGO. It's a ring-shaped protein that clamps onto the new RNA strand and chases after the polymerase. When it catches up, it uses energy from ATP to basically rip the RNA off the DNA template.
And the other way, it doesn't need a protein at all.
Right. It's called intrinsic termination. The DNA has a special terminator sequence. When the polymerase transcribes it, the RNA that's produced immediately folds up into a very stable hairpin loop.
And that hairpin physically yanks the RNA out of the polymerase.
It destabilizes the whole complex, and everything falls apart.
Okay, now let's jump to the eukaryotic world. The complexity level increases immediately. We have multiple polymerases.
3 of them in the nucleus. RNA polymerase 5 is a specialist. Its only job is to make the big ribosomal RNA.
RNA polymerase 7 is the real workhorse. It makes all the messenger RNAs plus a lot of the small regulatory RNAs.
And RNA polymerase 3 makes the other small functional RNAs, like the transfer RNAs and the small 5S ribosomal RNA.
And structurally, they're quite different from the bacterial enzyme.
They are. They're much larger with more subunits. And what's interesting is that their core structure is actually more similar to the polymerase found in archaea than in bacteria, which tells us something about evolution.
But the biggest difference is their total dependence on transcription factors.
It's night and day. Eukaryotic polymerases can't do anything on their own. They need a whole host of transcription factors to help them with every single step, from finding the promoter to starting, elongating, and stopping.
And for all 3 polymerases, the initial product is a long precursor molecule.
The primary transcript. It's the full-length copy of the entire transcription unit. And these pre-RNAs have to be processed, chopped up, modified, and assembled to become functional.
Let's dig into that processing, starting with the most abundant type of RNA, the ribosomal RNAs.
RNAs make up more than 80% of the total RNA in a cell. The cell needs a ton of it. So the genes that code for it, the rDNA, are present in hundreds of repeating copies.
And these copies are all clustered together in a specific part of the nucleus.
The nucleolus. The nucleolus is basically the cell's ribosome factory. It's not a membrane-bound organelle. It's more of a dense region where all the rDNA, the polymerases, and the assembling ribosomes are concentrated.
And we can actually see this factory in action, thanks to the famous Christmas tree micrograph from Oscar Miller.
It's one of the most beautiful visualizations in molecular biology. You're looking at a strand of DNA with an electron microscope. And along this strand, you see these repeating patterns that look exactly like Christmas trees.
The trunk of the tree is the rDNA gene itself.
And the branches are the nascent rRNA transcripts being made. The branches are short near the start of the gene and get progressively longer as the RNA polymerase I molecules travel down the DNA.
And the trees are packed so tightly together.
Incredibly dense. A new polymerase starts about every 100 base pairs. And you can even see little dots on the branches, which are the ribosomal proteins already starting to assemble onto the RNA while it's still being made. It's co-transcriptional assembly.
So the three big RNAs, 28S, 18S, and 5.8S, are all made as one single giant precursor.
A 45S pre-RNA molecule, about 13,000 nucleotides long. This whole thing then gets chopped up and heavily modified.
What kind of modifications are we talking about?
A lot. Over 100 methyl groups get added to the ribose sugars, and about 95 uridines get chemically converted into a different base called pseudouridine.
And these modifications are all in very specific conserved spots.
Which tells you they're critically important, probably for helping the RNA fold correctly or for guiding the cleavage reactions.
You can actually follow this process with a pulse chase experiment.
Right. If you feed cells a radioactive methyl group donor, you can watch the radioactivity first appear in the big 45S precursor in the nucleolus.
Then it moves into smaller intermediates.
And finally into the mature 18S and 28S rRNAs, which then get exported to the cytoplasm. It maps out the entire processing pathway.
And the cell has this amazing guidance system to make sure all those modifications happen at the right place. It uses other small RNAs.
The snoRNAs, or small nucleolar RNAs, they work as part of snor and P complexes.
And the snoRNA part is the guide. How does it work?
It's all about base pairing. The snoRNA has a sequence that's complementary to a specific spot on the pre-RNA. It binds there, forming a little RNA-RNA duplex.
And that duplex acts as a landing pad for the modifying enzyme.
Exactly. It physically brings the enzyme, say, a methylase, to the exact nucleotide that needs to be modified. It's an RNA-guided modification system. Incredibly precise.
So elegant. Okay, what about tRNAs?
tRNAs are made by RNA polymerase III, and Pol III is weird because its promoter is actually located inside the gene it's transcribing.
Not upstream like most promoters.
Right. And the pre-tRNA also needs processing. Its ends are trimmed off, and the enzyme that trims the 5' end is called ribonuclease P, or RNase P.
And RNase P is famous because it's a ribozyme.
It is. Its catalytic activity is in its RNA component, not its protein component, a major piece of evidence for the RNA world. And then just like RNAs, tRNAs get a whole bunch of their bases chemically modified.
All right, let's move on to the really complex story. The synthesis and processing of eukaryotic messenger RNAs.
This is where things get wild. The precursors to mRNA were first identified as heterogeneous nuclear RNAs, or hnRNAs.
Because they were, well, heterogeneous, all different sizes, many of them huge, up to 50,000 nucleotides long.
And they were only found in the nucleus. Pulse chase experiments quickly showed that these massive hnRNAs were the precursors that got processed down into the much smaller, mature mRNAs found in the cytoplasm.
Which explains a weird observation. If you label newly made RNA, most of the label goes into hnRNA, mRNA, even though they're a tiny fraction of the total RNA in the cell.
Because the RNAs and tRNAs are super stable and hang around for a long time, while the hnRNAs are processed and turned over very, very quickly.
So all of this is done by RNA polymerase II, and it needs that whole crew of General Transcription Factors to even get started.
The assembly of the pre-initiation complex, or PIC, usually starts at the TATA box,
a sequence about 25 bases upstream of the start site.
The first factor to bind is the TATA binding protein, or TBP. And when it binds, something dramatic happens.
It bends the DNA.
It bends it hard, about 80 degrees. This sharp bend acts like a signal, a landmark on the DNA that recruits all the other transcription factors and Pol II itself to assemble in the right place.
And one of those factors, TFIIH, has the enzymatic activities needed to kick things off.
Right. TFIIH is a helicase, so it uses ATP to unwind the DNA at the promoter, and it's also a kinase. It adds phosphate groups to the polymerase.
Specifically to a part of the polymerase called the carboxyl-terminal domain, or CTD.
This CTD is a long repetitive tail on the polymerase, and it's the master coordinator. When the polymerase first assembles at the promoter, its CTD is unphosphorylated.
Then TFIIH phosphorylates one of the serines, serine 5.
And that phosphorylation is the trigger. It allows the polymerase to escape the promoter and start transcribing.
But the story of the CTD doesn't end there.
Not at all. As the polymerase moves down the gene, other kinases phosphorylate another serine, serine 2. This changing pattern of phosphorylation on the CTD tail acts as a binding platform that recruits all the different RNA processing enzymes at the right time.
So the CTD physically links transcription with processing.
It's the ultimate multitasking platform.
Okay, so let's talk about that processing. First, the 5' cap.
This happens almost immediately when the new RNA is only about 25 nucleotides long. An enzyme adds a modified guanosine nucleotide to the 5' end, but it adds it on backwards in a weird 5' to 5' linkage.
And this cap has a few really important jobs.
It protects the mRNA from being degraded by exonucleases. It's required for the mRNA to be exported from the nucleus. And critically, it's the signal the ribosome looks for to start translation.
Then at the other end, we have the 3' poly A tail.
This happens at the end of transcription. There's a signal sequence in the RNA, AAU-AAA. An endonuclease recognizes that, cuts the RNA, and then another enzyme called Pol I polymerase comes in.
And it adds a long string of adenosine nucleotides, the poly A tail, without needing a template.
Right, maybe 250 of them. And this tail also protects the mRNA from degradation and plays a role in translation initiation.
But the most shocking discovery about mRNA processing came in 1977, the idea of split genes.
This just turned everything upside down. Everyone assumed that a gene was a continuous stretch of DNA that corresponded directly to the protein sequence.
But that's not true.
Not even close. The first evidence came from studying adenovirus. Researchers found that the beginning of the viral mRNA was actually pieced together from 3 completely separate chunks of DNA. The DNA in between was just gone in the final mRNA.
And this was quickly confirmed in our own genes using a technique called R-looping.
Right. If you take the mature mRNA for, say, the globin protein and mix it with the genomic DNA that contains the globin gene, they'll form a hybrid where they match.
But the DNA has a piece that the mRNA doesn't.
Exactly. So in the middle of the hybrid region, you see this big loop of single-stranded DNA sticking out. That loop is the piece that was cut out of the RNA. That's the intron.
The intervening sequence, and the parts that are kept are the exons.
And it turned out this wasn't an exception. It was the rule. The ovalbumin gene, for example, has 7 introns separating 8 exons. Most of the gene is actually intron.
Which means the cell has to perform an incredibly precise cutting and pasting job called RNA splicing.
And the precision has to be absolute. If you're off by even 1 nucleotide, you shift the entire reading frame for the rest of the protein and you get garbage.
So how does the cell know where to cut?
There are consensus sequences at the borders of the introns. A GGU at the 5' splice site and an AGG at the 3' splice site. There's also a key adenosine residue inside the intron called the branch point.
But in a huge gene with dozens of introns, those short sequences can't be enough to ensure accuracy.
They're not. There are also helper sequences called exonic splicing enhancers, or ESEs, that are located inside the exons. These sequences bind to proteins called SR proteins that help guide the splicing machinery to the right place.
And we know how important this is because something like 15% of all inherited human diseases are caused by mutations that mess up splicing.
It's a critical process, and the machine that does it is called the spliceosome.
This is a monster complex of proteins and small RNAs.
It's made of 5 snRNPs, small nuclear ribonuclear proteins. We call them U1, U2, U4, U5, and U6. And the spliceosome assembles fresh on every single intron.
Okay, walk us through that assembly.
First, the U1 snRNP binds to the 5' splice site. Then, the U2 snRNP binds to that branch point adenosine inside the intron, and it forces that A to bulge out.
It's preparing it for a chemical reaction.
Exactly. Then the other snRNP, U5, and the U4, U6 complex come in, and now a major rearrangement happens.
This is where it gets activated.
Right. U4, which was bound to U6, lets go and leaves the complex. This frees up U6 to base pair with U2, and it's this U6-U2 structure that forms the catalytic heart of the spliceosome.
And then the chemistry starts. 2 reactions.
The first reaction is that the 5' splice site is cut. The free end of the intron then attacks that bulging branch point A, forming a weird 2' to 5' bond. This creates a looped structure called the lariat intermediate.
It literally looks like a cowboy's lariat.
It does. Then the second reaction happens. The 3' splice site is cut, and at the same time, the 2 exons are joined together, the lariat intron is released and gets degraded.
And the fact that the catalytic core is made of RNA, the asin RNAs, strongly suggests that the spliceosome is a ribozyme.
That's the main takeaway. It's an RNA-based catalyst, although some recent evidence suggests a protein called PRP8 might be involved in the catalysis as well, so it might be a combined effort.
So why go through all this trouble? What's the evolutionary advantage of having split genes?
It's thought to be a relic of the RNA world. But more importantly, it allows for incredible evolutionary innovation through exon shuffling.
Because the introns are just spacers, you can have recombination happen anywhere inside them without messing up the coding parts, the exons.
Precisely. So you can take an exon that codes for a specific functional domain from one gene and shuffle it into a completely different gene, creating a new protein with a new combination of functions. It allows evolution to take huge leaps instead of just tiny steps.
And scientists have even shown this is plausible using test tube evolution.
They can synthesize billions of random RNA sequences and then select for the ones that can, say, catalyze a specific reaction. And they find them. It's proof of principle that RNA catalysts could have evolved naturally.
Let's shift gears to a more recent revolution in RNA biology: the discovery of small regulatory RNAs and RNA silencing.
This whole field started with a very weird result in petunias back in 1990. Scientists tried to make the flowers a deeper purple by adding extra copies of the purple pigment gene.
And instead, the flowers turned white.
Exactly. The extra gene was somehow silencing both itself and the original gene. It wasn't until 1998 that Fire and Mello figured out what was going on.
Working in the worm C. elegans.
They discovered that the trigger for this silencing was double-stranded RNA. They called the process RNA interference, or RNAi.
And it turns out RNAi is a fundamental defense mechanism. It's like a genetic immune system against things like viruses or transposons that make double-stranded RNA.
Let's walk through that pathway, the siRNA pathway.
So if a virus injects double-stranded RNA into a cell,
An enzyme called dicer finds it and chops it up into small 21 nucleotide fragments called small interfering RNAs, or siRNAs.
these siRNAs then get loaded into a protein complex called RISC.
The RISC complex then unwinds the siRNA duplex and keeps one strand, the guide strand.
And that guide strand is now the targeting system.
It is. The RISC complex uses the guide siRNA to find any messenger RNAs in the cell that have a matching sequence.
And when it finds one,
The key protein in RISC, called argonaute, acts like a pair of molecular scissors and slices the target mRNA in half, destroying it. It's a way to seek and destroy foreign genetic messages.
now compare that to microRNAs or mRNAs. These are not for defense, they're for regulating our own genes.
Right. mRNAs are encoded in our own genome. They're transcribed as a precursor that forms a hairpin loop structure.
And that hairpin gets processed, first in the nucleus by an enzyme called drosha, and then in the cytoplasm by dicer.
Dicer cuts it into the final 22 nucleotide mRNA duplex. This then gets loaded into a RISC complex, just like in siRNA.
But the mechanism of action is usually different, at least in animals.
It is. Animal mRNAs typically don't bind perfectly to their target mRNA. They usually only match in a small seed region in the 3' UTR.
So they don't trigger slicing.
Usually not. Instead, this imperfect binding either blocks the ribosome from translating the mRNA or it triggers the mRNA to be degraded through other pathways. It's less like an execution and more like fine-tuning the gene's output.
And the scale of this is enormous. Humans have over 1,000 different mRNAs that regulate a huge portion of our genes.
They're master regulators of development and cell function.
We should also briefly mention the Pol I interacting RNAs, or pRNAs.
These are a special class found mainly in germ cells, sperm, and eggs. Their job is to protect the genome of the next generation by shutting down transposable elements.
The so-called jumping genes.
Exactly. And their production is different. They don't use dicer. They're processed by a different class of argonaute proteins called PIWI proteins.
All of this just highlights how much of our genome is actually transcribed into RNA. The transcriptome is vast.
It is. We used to think most of our DNA was junk, but it turns out a huge amount of it is transcribed into non-coding RNAs, including many long non-coding RNAs that seem to be involved in regulating chromatin and transcription.
We're just scratching the surface of what all this RNA is doing.
It's a whole new layer of biological regulation that we're only beginning to understand.
Okay, let's move to the final act. Decoding the message. Let's talk about the genetic code.
Some of its properties were figured out logically even before it was cracked. First, it had to be a triplet code.
Because you have 4 bases, so a 2-letter code would only give you 16 combinations, which isn't enough for 20 amino acids.
Yep.
A 3-letter code gives you 64.
Exactly. Second, it had to be non-overlapping. When the ribosome reads the code, it reads 3 letters, then the next 3, then the next 3.
If it overlapped, a single mutation would change 3 amino acids in a row, which we don't see.
Right. A single base change almost always affects only a single amino acid. And third, the code is degenerate.
Meaning that most amino acids are specified by more than one 3-letter codon.
Right. 61 of the 64 codons code for amino acids. And this degeneracy is a key safety feature. If you look at the code table, you see that codons for the same amino acid are often grouped together.
And the difference is usually in the 3rd position.
The wobble position. This means that a random mutation in that third position often results in a synonymous change. It doesn't change the amino acid at all. The code is robust.
And the code was actually cracked in the lab using artificial RNA messages.
Nirenberg and Mathai's famous experiment, they made a piece of RNA that was just a long string of U's Pol I. They put it in a test tube with ribosomes and everything else needed for translation.
And out came a protein that was just a long string of the amino acid phenylalanine.
So UU must be the codon for phenylalanine. They did this with other combinations and eventually cracked the whole code.
And what's amazing is that this code is almost completely universal across all life on Earth.
A powerful piece of evidence for a single common ancestor.
So the molecules that actually do the decoding, that read the codons, are the tRNAs.
The adapters. These are small RNA molecules that fold up into a characteristic shape.
The two-dimensional view is a clover leaf.
Right, with several stems and loops. But in 3D, it actually folds into a compact L shape.
And one end of the L has the anticodon, the 3 bases that will pair with the mRNA codon.
And at the other end of the L is the 3' end, which always ends in the sequence CCA. That's where the amino acid gets attached.
Now, Crick's wobble hypothesis explains why we don't need 61 different tRNAs to read all 61 codons.
Right. He proposed that the base pairing rules are strict for the first 2 positions of the codon, but for the third position, there's some flexibility, or wobble.
So the same tRNA anticodon can recognize more than one codon.
Exactly. For example, if the first base of the anticodon is the modified base inosine, it can actually pair with U, C, or A in the third position of the codon. This makes the system more efficient.
But all of this depends on the first step being 100% accurate, charging the tRNA with the correct amino acid.
This is maybe the most critical step for fidelity. It's done by a set of enzymes called aminoacyl-tRNA synthetases.
And there's one synthetase for each of the 20 amino acids.
Right, and each enzyme has to recognize its specific amino acid A and D, all the different tRNAs that are supposed to carry it. It recognizes specific structural features on the tRNA to ensure it grabs the right one.
The reaction itself uses ATP for energy.
It's a 2-step process. First, the enzyme activates the amino acid using ATP. Then it transfers the activated amino acid onto the 3' end of the tRNA. The reaction is driven forward by the hydrolysis of pyrophosphate, making it irreversible.
And these enzymes even have a proofreading function.
They do. Many of them have a second active site, an editing site. If the enzyme accidentally attaches the wrong amino acid, it can move it over to the editing site, which will recognize the mistake and cut it off. It's a crucial quality control step.
Okay, we have our charged tRNAs. We're ready for translation. And it all starts with initiation.
And the whole point of initiation is to set the reading frame. The ribosome has to be placed perfectly on the initiation codon, AUG, so that it reads the rest of the message in the correct blocks of 3.
Let's start with bacteria. It's a simpler system.
It is. In bacteria, the small ribosomal subunit, 30S, binds to the mRNA first. And it knows where to bind because of the Shine-Dalgarno sequence.
This is a sequence in the mRNA, just upstream of the AUG, that base pairs directly with the 16S rRNA in the small subunit.
That pairing positions the ribosome perfectly. Then the special initiator tRNA, carrying formylmethionine, comes in and binds to the AUG in what will be the P site of the ribosome.
Then the large 50S subunit clamps on, and you're ready to go.
Right. This all requires several initiation factors, or IFs, and energy from GTT.
Eukaryotic initiation is much more complex.
It involves over a dozen EIFs. The big difference is how the ribosome finds the start codon.
It doesn't use a Shine-Dalgarno sequence.
No. Instead, a complex of initiation factors binds to the 5' cap of the mRNA. Another factor binds to the poly A tail. And then these two ends are brought together, effectively making the mRNA into a circle.
Why circularize it?
It's a quality check. It ensures the ribosome only translates full-length, intact mRNAs that have both a cap and a tail.
So once the mRNA is circularized, the small 40S subunit, already carrying the initiator tRNA, binds to the cap.
And then it starts to scan. It moves down the mRNA using ATP for energy until it finds the first AUG in a good sequence context. That's where it stops.
Then the large 60S subunit comes in, the initiation factors leave, and you're ready for elongation.
And the structure of the ribosome itself is amazing. It has 3 sites for tRNAs.
The A site for aminoacyl-tRNA, where the new tRNA comes in.
The P site for peptidyl-tRNA, which holds the tRNA attached to the growing polypeptide chain.
And the E site for exit, where the empty tRNA leaves.
And the most important part is that the catalytic core, the peptidyltransferase center that actually forms the peptide bonds, is made entirely of ribosomal RNA. The ribosome is a ribozyme.
Okay, let's walk through the elongation cycle.
Step 1. A new charged tRNA bound to an elongation factor in GTP enters the A site. This is a fidelity check. Only a tRNA with the correct anticodon will lock in properly and trigger GTP hydrolysis.
Step 2. Peptide bond formation.
The peptidyltransferase ribozyme catalyzes the reaction. The growing polypeptide chain is transferred from the tRNA in the P site to the amino acid on the tRNA in the A site.
Step 3. Translocation.
The entire ribosome now moves exactly 1 codon, 3 nucleotides down the mRNA. This is a huge mechanical movement.
It involves a ratchet-like motion between the two subunits.
Right. And it requires another elongation factor, EFG, and another GTP hydrolysis. This movement shifts the tRNA that was in the A site into the P site, and the empty tRNA that was in the P site gets moved to the E site.
For where it is then ejected, and the A site is now open for the next tRNA.
And the cycle repeats, adding 1 amino acid at a time.
Until the ribosome hits a stop codon.
Right. UAA, UAG, or UGA, there are no tRNAs that recognize these codons.
So instead, proteins called release factors enter the A site.
These release factors mimic the shape of a tRNA. They recognize the stop codon and trigger the ribosome to cut the finished polypeptide chain off of the last tRNA. Everything then dissociates and is recycled.
Now there's one last quality control system we need to talk about, and it links splicing to translation. Nonsense-mediated decay, or NMD.
This is a surveillance system to get rid of mRNAs that have a premature stop codon, which can arise from a nonsense mutation. These mutations are responsible for about 30% of all inherited diseases.
So how does the cell know a stop codon is premature?
It uses the memory of splicing. When an intron is spliced out, the spliceosome leaves behind a little protein marker called the exon junction complex, or EJC, on the mRNA.
So a normal mRNA will have these EJCs scattered along its length.
Right, and as the first ribosome translates the message, it plows through and knocks off all the EJCs.
But if there's a premature stop codon,
the ribosome will stop and fall off early, leaving any downstream EJCs still attached to the mRNA.
And it's the presence of those leftover EJCs that signals to the cell, hey, this mRNA is defective.
And the NMD machinery is recruited, and the faulty mRNA is rapidly destroyed before it can make a truncated, potentially harmful protein. It's an incredibly clever system.
And finally, to maximize efficiency, you often see many ribosomes translating the same mRNA at the same time.
A polyribosome, or polysome. In bacteria, this is even coupled with transcription. You can see ribosomes hopping onto the mRNA while it's still being synthesized by the RNA polymerase.
Okay, let's finish up by looking at some of the practical applications and key experiments highlighted in our source material. First, the clinical potential of RNA interference.
The big dream here is to use synthetic tRNAs as highly specific drugs as magic bullets to target and destroy the mRNA from a cancer-causing gene or a virus.
The problem has always been delivery. How do you get these fragile RNA molecules to the right cells in the body?
This is where nanotechnology comes in. Researchers are designing specialized nanoparticles to carry the tRNA payload.
Like the experiment for treating colitis or inflammatory bowel disease.
Exactly. They packaged an tRNA into liposomes, and they coated those liposomes with an antibody that specifically recognizes a protein on the surface of inflammatory white blood cells.
So the nanoparticles would home in on the problem cells.
They did. They delivered the tRNA, which shut down a key pro-inflammatory gene, and it dramatically reduced the disease in animal models. It shows that targeted delivery is possible.
And let's circle back to that foundational discovery, RNA as a catalyst.
This really shattered the old dogma that only proteins could be enzymes. It started with Thomas Sick in 1981, studying rRNA splicing in the organism Tetrahymena.
He was trying to purify the protein enzyme that did the splicing.
Right, but he found that if he just took the purified pre-rRNA and put it in a test tube with some ions and GTP, no protein at all, it would splice itself.
The RNA was its own enzyme, a ribozyme.
It was. And then a couple years later, Sidney Altman and Norman Pace showed that the RNA component of the enzyme, RNase P, could cut a separate tRNA substrate molecule.
So it wasn't just self-splicing. An RNA enzyme could act just like a protein enzyme on other molecules.
And that discovery was the final crucial piece of evidence that gave real weight to the RNA world hypothesis.
So we've really covered the entire journey, from Garrod's first inkling that a gene makes an enzyme, all the way through the intricate dance of the spliceosome and the ribosome.
It's an incredible story. And the key takeaways are just how central RNA is to everything. Its complex structure, its catalytic power in splicing and translation.
And the discovery of that whole new world of tiny regulatory RNAs the mRNAs and tRNAs, that add this massive layer of control on top of everything else.
I think the most profound thing to sit with is just how ubiquitous RNA is. It's the messenger, the scaffold, the adapter, the regulator, and the catalyst. The ribosome, the engine of all life, is fundamentally a ribozyme.
Which leads to a really interesting final thought. If the core of protein synthesis and splicing are run by RNA, what other fundamental cellular processes that we've always assumed are run by proteins might have an ancient RNA machine hidden at their core, just waiting to be discovered?
That's the question that keeps the field exciting.
Food for thought. Thank you for joining us on this deep dive.